Hey guys, Miss Jano here. So I'm here to explain to you the structure of DNA. You guys should already have the notes of this PowerPoint because you were made to copy this last week. I just want to clarify in case you have any questions. You can always reach me, and I'm just going to go over the PowerPoint so that you guys can understand maybe a little bit better, okay? So the first thing that we have to cover is the essential vocabulary. The first word that you have to know is double helix, okay? And that means that that is the twisted shape of a DNA and it resembles a twisted ladder. So this right here is an example of a double helix. And the first thing that I want you to notice is the orange backbone. So I'm tracing that over with the red right now. Imagine that this was like two sticks of a Twizzler candy and you wrap them around. That is basically the shape of DNA. So that is a double helix. Why is it double? Well, because you have two strands that are wrapping around, okay? The second word that you should know is nucleotide. I want you to make sure that you remember that nucleotide has a T, and we're going to see why that's important in a second. So a nucleotide is a subunit of a nucleic acid molecule that consists of a phosphate, a sugar, and a nitrogenous base, okay? So keyword right here is subunit. If you are a subunit of something, you are not the full thing, but you are a piece of that thing, okay? So a nucleotide is a subunit of a DNA molecule and it consists of three things. So this is why this is important because there's a different word that is very similar to it, okay? So I want you to think of nucleotide, the T in nucleotide, think of it as for three, okay? So that consists of a phosphate, a sugar, and a nitrogenous base. And then the second word that is part of the essential vocabulary is nucleoside. And this is also a subunit of a nucleic acid molecule, but this one only contains two things, a sugar and a nitrogenous base. So this one only has two. So if in the future I ask you what is the difference between a nucleotide and a nucleoside, the biggest difference is that one has a phosphate and the other one doesn't, okay? So a nucleotide is technically the bigger subunit, okay? And then the nucleoside is the smaller one. So also look at this image right here. Notice that the phosphate group is not part of this little bracket. So I'm cutting that off basically. A nucleoside has the little hexagon, which is your nitrogenous base. This thing right here. Oof, excuse my drawing. And then it has the pentose sugar. Again, excuse my drawing. Okay, but I do want you to notice something. The sugar is always going to be connected to the phosphate group and then to a nitrogenous base. The nitrogenous base can change, but the sugar is always going to be connected on one side to the phosphate group. Okay, so remember that nucleotide has three components and nucleoside has two. What is the biggest difference? The biggest difference is this phosphate right here. Nucleotides have them, nucleosides do not. Okay. So what is a monomer of DNA? I think this is a key word right here because you guys have been exposed to this in the past. Remember that a monomer, mono equals one and poly equals many. So if you have a polymer, you would have many parts of DNA. But if you have a monomer, you have only one. So this right here is a single monomer of DNA. So it's only one part of DNA. Okay. And a monomer of DNA is a nucleotide. So you already know this word. So I'm going to put this right here, nucleotide. And if you remember, is a nucleotide made up of two or of three parts? So remember that because it has the T, think of three. So it's made up of three parts. Okay, so what is this circle thing right here? What is the little pentagon? And then we're going to get to what this big thing is in a second, okay? So if you look to the picture on the left, you'll notice that the little blue balls are all connecting each other through the pentagons that are the squares, okay? So what this is right here, if you remember from the previous picture, this is a phosphate, and that is always going to connect to a sugar, and that sugar is always going to be connected to a nitrogenous base. Okay, so if we go back for a second, remember that the components of a nucleotide, which were three, are nitrogenous base, sugar, and a phosphate. A phosphate and a sugar are always going to be the backbone of DNA. The nitrogenous base can change, and you're going to see what are the 4 options that can change. Actually, you can see them here in the picture. So you have A, G, C, and T. And for those of you that don't know how to pronounce that, that's adenine, guanine, cytosine, and thymine. Okay, but we'll get to that in a second. Oh, the pictures are already there. Okay. So what are the components of a DNA molecule? Here you go. So we know for a fact that the backbone is always going to have these 2 parts, the sugar and the phosphate group, okay? And if we go back for a second, you're going to notice how the phosphate group is right here, the circle. So from now on forward, if you ever get a similar image to this, know that the phosphate group is the circle, okay? So the phosphate group is the circle, and the sugar is always going to be almost like a little house, basically. So this is where things get interesting, and this is where you should pay attention, because this is where the work for this week is basically going to be reliant on, and that is the nitrogenous bases. You can have 2 kind of nitrogenous bases. You can have pyrimidines and purines. These guys are different chemically, but they bond with each other. So a T is always going to be connected to an A, and a C is always going to be connected to a G. So for example, if you have let's say if you have a molecule that is GCA, you know that the G, this one right here, is always going to go with the C. If you start out with a C, well, who do you pair with? A G. And if you start out with an A, well, then who do you pair with? A T. So they really only have a partner, a perfect partner. So A with T and T with A, C with G and G with C, okay? And that's what you're going to practice basically this week for homework, okay? So what is the overall shape of DNA? The shape of DNA resembles a twisted ladder or double helix. In this example right here, nothing is twisted so that you can see the simplistic breakdown of it. So the backbone is formed by a sugar and a phosphate, and you've already been exposed to that. And that is part of the nucleotide. Remember that the nucleotide has something else that is included within it. But then this nucleotide also includes something called steps. So the steps of a ladder. And these are the things that are made from nitrogenous bases, and they stick out sideways. So go to the picture now and notice how here you have your little house. And like I said, that little house is always connected to a phosphate. But then again, who's that new phosphate connected to? Oh, a little house. And then again to a phosphate and a little house. And basically like that. Again, I'm sorry for the drawings. They're really ugly. Okay, so the phosphates and the sugars, which are this right here, the stuff that's not in pink, are always going to be part. So this right here is your backbone, okay? You can't have a DNA if you don't have this. However, where do you store genetic information? So what is the meat and potatoes of this stuff? And that is the letters that are in pink. Those are what you would call the steps of the ladder, so the steps of the DNA double helix, and these are the ones that you were just exposed to a second ago on the previous slide. So you have adenine, thymine, guanine, and cytosine. So who does C always pair with? G. And who does T always pair with? A. It's not possible to have a T pair with a C or a G, okay? So that's never going to work. So you always have to remember that if you start out on one side and you have an A, it's always going to be complemented by a T on the opposite side, or vice versa. And the same thing with G and with C, okay? So these are the steps, and these are the actual portions of DNA that contain genetic information. Now, who's to say that instead of having a G here, I could have had a C, and instead of having a C here, I could have had a G. The fact that I just changed the arrangement of one of the DNA backbones means that now I cannot have a C here. What letter do you think goes here? It can't be T and it can't be A. The only other one could be G. And now if I have a G on this side, who do you think goes on the opposite side? Well, it's a C. And if I were to have another C here, well, instead of having an A, here I would have a G. So it all depends on who you have on one side to then be able to find the complement. And we all look different. We all have different features. So the order in which these letters are is different for each and every one of us, okay? So let's assume that this would be your number 1, your number 2, your number 3, and your number 4. I could have this arrangement, but somebody else could have a different arrangement, okay? So this is all different for everybody. The only thing that never changes is who your base pairs connect with. So A is always going to connect with T, and T with A, and G is always going to connect with C, and C with G, okay? So as it says here, the nucleotides can be joined together in any order. So that means that any sequence of bases is possible, which is why you and I look different. So that is why we all have different genetic information, okay? This right here is just a little background information as to how we've come across all of this DNA research. You can read that on your own. And this pretty much is the end of my video. Replication of DNA is what we're going to be covering next week, okay? So stay tuned.